All right. Everyone ready to get started?
All
Excellent.
right. Well, welcome everybody to Paleo Talks. We are at episode 23. And this week we're going to talk with Dr. Leah Lynch. Hey, Leah.
Hello.
Great to have you.
Thank you. Thanks for having me.
We'll do more introductions with Leah in just a second. And we also have Chris Widgo with us today. And Chris is available on the screen. He's going to start hanging out with us to ask some more questions as well. Make sure we don't have any gaps in talk. So between the four of us, we should have lots of talk. And David, we'll go over you for just a second so you can do a quick summary of how the show works again.
Sure thing. As usual, we are going to start. Once we've introduced our guest, we're going to let Leah go ahead and give a presentation today talking about pine marten evolution, which, as we were discussing earlier, will make this one of the cutest in our Paleo Talk series. We're going to, as Leah goes through the presentation, about halfway through, so after about 30 minutes or so, we're going to start turning it over to audience questions. At that point, the audience, if you're here on Facebook, you can go ahead and start putting your questions in the comments section of the Facebook video. And as usual, if you can't for any reason leave comments on Facebook, go ahead over to the Gray Fossil site, Twitter or Instagram, and send us your questions there. I'll be keeping an eye on everything, so hopefully we'll be able to get through as many questions as possible.
All right. Well, we are again coming to you from East Tennessee State University and the Center of Excellence in Paleontology, which oversees the Gray Fossil site here in Tennessee. And we always sort of like to relate that Gray Fossil site to our presentation of the day. And Leah was actually a student of ours a few years ago. And we'll just sort of jump over to Leah now and let her tell us about her history in becoming a paleontologist, which includes coming here to East Tennessee State University.
Yeah, so I'm one of those kids who got obsessed with paleo at like age 8 and then never looked back. So I went to Bowling Green State University and worked under a wonderful invertebrate paleontologist, Peg Yakabuchi. And I got a master's at ETSU and worked with Blaine and Wally there, and was actually in the same cohort as David. So we've known each other for a while. I then went to Oklahoma State University, where I got my PhD and kind of switched gears a little bit in the sense that I now teach anatomical sciences. So I have been working at med schools for the past many years, 8 years, 7 years, I don't know, for a while. And so as a paleontologist, that's actually a pretty common route. A lot of us, since we know our anatomy and we study the anatomy of animals, we can then teach gross anatomy. So after finishing at Oklahoma State, I went to WashU and I did 2 years of postdoc. And now here I am as an assistant professor at Midwestern University in Glendale in Phoenix.
And how long have you been there?
Like a month and a half. I'm very new.
Well, thanks.
How long have you physically been there?
Yep.
Oh, since like June. So still very new.
Right.
Thanks for pointing out the job aspect. And it is true. A lot of people who go through and end up doing paleontology in their career end up working, teaching medical students. And I think a lot of folks don't realize that's a potential job for paleontologists.
Yeah, and there's a lot of great programs, great PhD and master's programs in those schools. So definitely worth checking out.
All right. Well, again, Leah is at Midwestern University in Glendale, Arizona, and she works on a wide variety of carnivorans. And today's going to talk to us about pine martens, which is something we used to have right here in the Appalachians of Tennessee. So I know she's going to tell us a little bit about their past record and where they're at today. So if you want to pull up your presentation and get started, that'd be great.
All right, you guys seeing that?
Mm-hmm.
All right, okay, so I will start rolling with it then. So, what? I don't know. Hang on. What are we doing here?
Working for you?
Yeah, I tried to minimize too many windows. Let's see if that works now. There we go. Okay. So what I'm really interested in my research is understanding how differences in climate and biome can act as selective pressures and then influence the phenotype or shapes of bones or color of animals or any other like morphological trait. And then I'm also interested in the evolutionary processes that end up making these traits come about as they are. So things like genetic drift, phenotypic plasticity, or adaptation through natural selection. And I'll kind of give examples of all of these as we go. But just to kind of give you a background, the animals that I work on are predominantly found in the Pleistocene. And so the Pleistocene, even if you don't know it by name, is a time period that all of us are familiar with because it has a lot of charismatic animals in it, right? We have mammoths and woolly rhinos and saber-toothed cats and bison and lions and all sorts of awesome big animals. And the Pleistocene ranged from about 2.5 million years to 11.7 thousand years ago. And it's also really well known for its glaciers. So most of North America, Europe, and Asia was covered in these big glaciers for extensive periods of time, but they weren't consistently there. So instead what happened is there were cycles of these glaciations where glaciers would expand across North America, for example, like you're seeing here, and then they would retreat, and then it'd expand and then retreat. And so every time that this happened, you think about these big ice sheets pushing across Canada, right? You would lose all of that habitat. And then as they retreated, all this new habitat would develop. And it might not be the same as the habitat that had been there previously. And so what glacial cycles do is they have this fluctuation in climate, they're reorganizing biomes, and it presents what we call novel selective regimes. So in other words, a new suite of characters that animals have to respond to in order to be able to survive in that new habitat. But at the end of the Pleistocene, not all of our mammals were successful in surviving that last change into the modern biomes that we see today. So a lot of us are really familiar with our megafaunal extinctions, right? In North America, we lost the American lion, which I'm particularly partial to. We lost mastodons and horses and giant ground sloths. And so we saw this big die-off, but really what I'm more interested in are the animals that didn't die off. So there's a lot of animals in North America that not only survived these changes, but in climate and biome, but we've shown that they underwent some form of genetic evolution or phenotypic evolution as they were shifting into these new biomes that we see today. And so some of these animals include like the yellow-rumped warbler that you're seeing here, different species of tree squirrels, voles, gray wolves, mountain goats, bighorn sheep. And what's kind of cool about this is they all share this exact same geographic range, this green range here on this map. And so that made me wonder whether other animals in that same range also underwent some form of phenotypic or genetic evolution as glaciers retreated and they were expanding into this new biome and habitat.
Leah, can you remind the audience what a phenotype is?
Yeah, so a phenotype is like a trait, right? So if I'm describing like the phenotype of my hair, I would say that it's like a brown red. So it's going to be like a characteristic that that animal is exhibiting. So the animals that I decided to kind of hone in on to understand evolution in the Pleistocene were the mustelids. So mustelids are carnivorans, so they're related to like bears and dogs and hyenas and cats. And Mustelidae is comprised of animals like otters, ferrets and weasels, martens, like you see here, and then badgers, just as a few examples. And so the animal that I really looked at was Martes americana, which I've highlighted here in purple and is the adorable little guy depicted there, the third picture down. So North American pine martens share one common name, but there's actually two different species, Martes americana and Martes carina. And they're all pretty closely related to animals that live in other continents, actually. So the European pine marten, Japanese martens, but also wolverines and fishers. And fishers are here in North America as well as wolverines. So the reason why they share the same common name, why we say, you know, American pine marten, even though there's two species, is because we actually didn't separate them out into two different species until really recently. So there's been several studies that use kind of a combination of evidence to support that these are two different species. So what you're seeing here is an evolutionary tree looking at several individuals of Martes carina and Martes americana. And you can see that there's like two big clusters there supporting that they are not related to one another as a single species. But you can also see in the picture here that their cranial shape differs, right? So we're looking at their skulls, and they're quite a bit different from one another. Martes americana is more elongate, Martes carina is a little bit more robust, but they also have similar parasites. So they actually use parasitological evidence to support that they would be separate species because they looked at the genetics of the parasites, which is pretty cool. So we find Martes americana and carina in kind of two different regions in North America today. So Martes americana is depicted here in this turquoise teal color. It's found in northeastern United States, most of Canada, and Alaska. But Martes carina is really restricted to the western US and western parts of Canada. And so throughout the rest of my talk, I'm going to do what David suggested and make this the most adorable talk you guys have ever had. So here's our pine marten, and they're going to be color coded based on whether I'm referring to Martes americana or carina throughout the rest of the talk, okay? So throughout their geographic ranges, these two species are subjected to a pretty wide climatic variation. So anywhere between 19 to 200 centimeters of rainfall, 17 to 614 centimeters of snowfall, and temperatures ranging from -35 to 29 degrees Celsius. But they also have pretty different biomes that they occupy. So Martes americana is found in boreal forest biomes as well as temperate mixed broadleaf forests, whereas Martes carina is predominantly in coniferous forests. And so they have a wide variety of diets then too, because there's going to be different animals living in each of these biomes. So they pretty much, I like to think of it as they'll put anything in their mouth that they can fit in their mouth. Some of the diet studies have even found rocks. So, you know, whatever they can catch, I guess. But they'll eat things like birds, amphibians, berries, and different mammals. And depending on which biome they're in, they have a different dominant prey. So Martes americana might be hunting squirrels predominantly in boreal forests, but carina will be hunting voles in coniferous forests.
So is the pink the Pleistocene distribution?
So the pink is showing there the biome distribution. Yeah, but in the Pleistocene, that is where we would have found them. Yeah, and I'll show that in a second. Yep. So because they have such a wide variety of diets and they differ in their predominant prey, they also then exhibit different hunting behaviors. So animals that are in boreal forests are really good climbers, and they're all capable of climbing, but you see a lot more climbing in animals that are hunting squirrels, obviously, like this guy. Whereas in regions where they're experiencing a lot of snowfall or they're hunting voles, they're navigating like tunnel systems that these small mammals have created underneath the snow layers. So they're hunting differently in these different environments as well. And so to top this all off, this is really starting to suggest that, you know, maybe we're going to see differences in morphology because they're behaving differently and they're experiencing, you know, different climates and biomes in their different regions. But our fossil evidence also supports that they were separated from one another at least once during the Pleistocene. So these circles here are depicting where we would have found Martes americana and where we would have found Martes carina. So Martes americana used to actually be found during the last glacial maximum when all of Canada and parts of northern United States were covered in ice. Martens were found in the Appalachians, whereas out west, they were found in the Rockies and the Sierra Nevadas. And that's where we predominantly see them today too. And so this kind of led me to wonder like whether or not glaciation would have affected their evolution. And so I decided to answer this question by looking at their limb morphology, because they're moving around with that, they're, you know, hunting, they're experiencing the environment that they're tactile touching. So I thought that would be the most informative signal. So what I did was I tried to test whether or not their limbs were an adaptation to biome and climate. But in order to do that, I first had to determine when they would have diverged genetically from one another. So when did we actually see that first split between the two species? And then whether or not there was any statistical relationship between the skeletal morphology and then climate and biome traits. So let's focus in just on the genetic part first. So in order to create a phylogeny or an evolutionary tree, right, depicting our relationships, I first have to get DNA out of these animals. I then use the information from fossils to calibrate the tree, and then I can generate the phylogeny. So I'm going to walk through each one of these. So in order to get DNA from samples, I reach out to museums and I ask them for specimens of martens. And so a lot of museums have in their collections either soft tissue or skeletal samples of pine martens. Most of the soft tissue are things that have been collected since the 1990s. Anything older than that is usually just skeletal. And so they may give me something like liver tissue or cardiac muscle, or I'll get bone or like a rib, for example, or a tooth. And then what I do is within the lab, depending on the tissue type that I'm working with, I have a little bit of a different process for extraction. So if I'm working with soft tissue, I can basically put it in a vial, subject it to some chemical and enzymatic processes that will break down the cells, they break down the nucleus, and then they allow you to just separate the DNA out. And I get to wear ridiculous outfits like this. I predominantly wear this when I'm working with things that are older than like the 1970s because their DNA can be a little bit degraded. So it's not in beautiful long chains, it'll be in kind of pieces. And so I want to make sure that I'm not contaminating them with my own beautiful DNA. And so I'll wear things like this. And then after the whole process, then we have Marten DNA. So what I then look for time calibrating is I use wonderful resources like the paleobiology database here. And so what you can do on this free resource that people have put blood, sweat, and tears into pouring different fossil sites and information into is you can look up a species and see where fossil sites of that species have occurred. And then you can see the age of those sites. So I can use that time calibration data to inform my tree. So essentially, I take my genes and I align them. So you end up with something that looks like this. So all the A's, C's, G's, and T's are all in line with one another. But you can see that there's points where, you know, most of the animals here have a T, but then some of them have a C. So as the analysis is running, it's looking for those similarities and differences, and then it's going to start pulling them apart based on those. But you can inform the analysis then with things like the how genes evolve, right? If a C is more likely to turn into an A, or if it's more likely to turn into a G, you want to kind of give the analysis a little bit of information about that. You can put in molecular clocks, so a lot of us are familiar with that, which is kind of like the rate at which a gene would be evolving or nucleotides would be changing. And then based off of those fossils, ages, I can also put in estimates of minimum or maximum ages that I would expect to see different branches occurring. And you don't set that, you don't say like, I want this node to be that exact time. You give it in there as a like range, and then the analysis bases that as kind of like an informed prior. So what you end up with is a tree that looks like this. So this is from one gene. This is from the cytochrome B gene, which is in the mitochondrial genome. All of the California 15 or whatever names that you're seeing on the right are all individuals that I sequenced. And then the ages on here are informed from those fossils. And so I was able to determine that we're seeing that split between Martes carina and Martes americana sometime around 540,000 years ago. And the super cool thing you can do with this, since you know, or you have at least an estimate of the time at which this took place, is you can put that in an environmental context, because we know when glacial cycles were taking place. And so you can, what I've done here is I've overlaid the different glacial and interglacial episodes onto the phylogeny. And so what you can see is that Martes americana and carina split during one of these blue bars, so in the middle of a glacial period. And that those clades were then diversifying also in the middle of another glacial period. And so this really gets me thinking that, okay, maybe glaciers are playing a role in this and separating them out from one another. And so if they were splitting around 540,000 years ago, then both fossil and now the genetic evidence is supporting that they were isolated from one another from glaciers spreading across Canada at least 6 different times. So they came apart, they came back together, they came apart, and that continuously happened until we have the populations that we see today.
Leah, do you have any idea whether or not they will still breed and produce viable offspring if together?
Yeah, they do. Yeah, and that gets into my theories a little bit later too.
Okay.
Yeah. So next I wanted to look at their limb morphology and whether or not that correlates with their climate and biome. So here's where my morphological studies come into place. So I measured the limb morphology of about 70 different individuals. The green points here representing where I collected them from, or where the museum collections collected them from. And then I compared it to different climate data from the county and the year that these animals were collected. So like snow, rainfall, and temperatures from those regions. And so I had kind of 2 forms of data that I was looking at here. I was interested in the shape of the bones as well as limb proportions. So how long the forelimb is relative to the body size, or how long the hind limb is relative to body size. And so for that, I just used linear measurements. So in order to get shape data, I use geometric morphometrics. So this is a pretty common practice now to get shape information from bones. For this study, I used a microscribe digitizer, which essentially is like taking a little stylus and putting it on a bone, and then you record an XYZ coordinate. And then all of those coordinates come together to kind of represent the shape of that bone. Nowadays, some things that other people are doing is they're using like CT scans or surface scans of bones, and then they're putting landmarks on that way, but you're still getting an XYZ coordinate. And so in order to compare then all of those coordinates to one another, you have to run something called a generalized Procrustes analysis, which essentially takes those landmarks from all the bones, it rescales them, it rotates them, and it overlays them so that you can directly compare each individual to one another. And you get a plot that kind of looks like this. So all those little like blobs are the points from individual specimens where that landmark would have been. But you're working with then a ton of data, right? So if I have 12 landmarks and I have 3 coordinates, the XYZ coordinates for each one, then I have 36 variables in my study. And that's a lot. So we often run things like a principal component analysis, for example, to kind of reduce that dimensionality down so you're working with 1 or 2 variables instead. So this basically takes all your points and it plots them in like 36 dimensional space and tries to fit a line to that, a best fit line. And then you can end up with plots that look like this that are kind of compressed down. And you can use those single variables then in statistical analyses. So that's what I do then when I'm comparing between groups. So you can also visually compare these using things like thin plate splines. So this essentially is taking a grid, overlaying it onto the bone morphology, and then it warps those grids into one another and gives you kind of what's called a deformation grid. And so you look at kind of boxes on this grid that aren't a box anymore, and that tells you where landmarks have shifted or where differences in shape are occurring on the bone. So kind of using all of those things together, I ended up finding that there were differences in limb shape among animals that lived in broadleaf forests from coniferous. Boreal forests differ from coniferous, and then boreal and broadleaf differ. And this is really cool because boreal forests and broadleaf forests, those are the same species, but their morphology differs across the different biomes. And what we saw here was that the animals living in these broadleaf forest biomes have a much more gracile morphology, so they're a little daintier, if you will, whereas those living in coniferous forests are really robust. If we look at forelimb proportions, we also saw differences among biomes. So the proportions in broadleaf forest animals was different from the other 2 biomes. And in the hind limb, it's actually a different pattern, which is also really interesting, where the coniferous forest animals have different limb proportions in the hind limb than those from boreal and broadleaf forests. So these shapes also correlate with climate. So we're seeing a correlation with biome, and there's also now a correlation with climate. So animals living in regions with high snowfall are much more gracile. So perhaps having that dainty morphology allows them to walk around on the snow or underneath the snow a little bit easier and not, be so heavy or falling through. But then the opposite is true of rainfall. So animals with gracile morphologies live in regions with less rainfall. So perhaps that has to do with vegetation density, since that's dependent on the amount of rain that they're getting. Limb proportions also correlated with climate variation. So different animals that have long limbs tend to live in regions with high rainfall, but they are living in regions with low temperatures. So there seems to be a relationship happening here. So one, we have these animals diverging in the middle of a glacial cycle around 540,000 years ago. And nowadays, their morphology is correlating with their climate and biome. So this really got me back then to my glacial cycles and saying, okay, if we're seeing these relationships with biome and climate now, it's probable that they evolved in relation to biome and climate in the past. And that if they evolved in the middle of a glacial cycle and throughout several glacial cycles, then those probably influenced the evolution of their morphology of their limbs. So that gives me back then to understanding if these are acting as selective pressures, biome and climate, then what are the evolutionary processes that drove us to see the variation that we see today? So what I do with a combination of my data then is what we call phylogenetic comparative methods. So I'm basically modeling different evolutionary processes. And so to do that, you need a phylogeny, which we have based on our genes that we extracted from individuals. And you also need a morphological trait. Now, the key here is that I'm using the phylogeny and the morphological traits from the exact same animals. So they're directly comparable to one another. And so with that information, you can answer questions like how a trait evolved, the rate at which it was evolving, if these traits are more similar in animals that are genetically related to one another, or if that doesn't matter. And then you can also look at how disparity, or the amount of variation in a population, has changed through time as the species evolved. And so what I did was then I sampled the DNA and morphology from the specimens that you see highlighted here with the little colored bars. And so you can see I fairly evenly sampled individuals from each of the biomes within our phylogeny. And then I, again, looked at shape data and size data, but in this case, I used size as the centroid size. So this is a measure that's coming from our landmark data and is basically telling you the overall size of the bone. So it's not the same as like a linear measurement, but it's giving you kind of a roughly similar idea. And so then using those traits and then using my phylogeny, I sought to answer these questions on how they were evolving, the rates, things like phylogenetic signal, which is telling you how closely related individuals are, whether their traits are similar. So I'll just kind of skip over all the technicalities of that and give you the cool stuff. What I ended up finding was that the shape of these limb bones is really reflecting their underlying evolutionary relationships, which means there's a strong phylogenetic signal. So two individuals that were genetically closely related to one another had very similar morphologies. But their rates of evolution of shape is actually pretty slow. But when we look at size, we're looking at punctuational. So every time there's a branch on the tree, we're seeing a change in our morphology and our size of our limbs. But there's low phylogenetic signal, which means these changes are happening, but it doesn't necessarily mean that two individuals that were closely related are looking more similar. They could actually be very different from one another. And they're evolving much faster too. So what I took away from this, the super cool thing, is that that means that the shape of these limb bones and the size of these limb bones is evolving differently. So they're almost independent of one another and they're being selected on differently, which is super cool because it's one bone, right? You'd think it all just goes along in a package, but it's clearly not. And so what I kind of took away from this is that means that our biome and climate must be acting as selective pressures. If we're seeing these correlations, we're seeing these two traits evolving differently, then they must be acting as selective pressures, but perhaps differently on shape versus size. So perhaps climate is affecting size and biome is affecting shape, or something along those lines. So what we think is happening here. So I should throw out too that this portion of my research I did in collaboration with two awesome paleontologists, Dr. Haley O'Brien and Dr. Ryan Felice. So if you're interested in this kind of stuff, they've got some awesome papers too. So we think that what this is showing us is that limb shape has evolved as an adaptation. So here I have my original population of martens, say, and their genotype is coded here in A's and B's, and their phenotype are the colors of their legs. So if they're subjected to a selective pressure, say like an increase in temperature, then some of those martens might not do particularly well. And so those that survive, they have increased performance, increased fitness, they carry these genes off to their offspring, and they end up then with our resulting population having a different genotype and phenotype. So we start out with one population whose mean phenotype and genotype is green legs and a bee, but our resulting population is different because of that selective pressure. But limb size seems to be doing something completely different and doesn't seem to have that same genetic component. So what we think is happening here is that we're looking at something that's more phenotypically plastic. In other words, say I have my population here, you'll notice that they have different phenotypes, but that all of them have the same genotype, right? They're all A's. So that means that that A genotype can produce multiple phenotypes, and it does so across an environmental gradient. So all my green legs seem to do really well in regions that are cold, and all my pink legs seem to do really well in regions that are warm. So say my cold regions are in my broadleaf forests and my warmer regions are in my coniferous forests. Well, if for some reason coniferous forests suddenly were to get colder, then I would see a shift in the phenotype, but my genotype would stay exactly the same. And so what we see there is then a difference in our phenotypes changing, but the genotypes stay the same because of that change in temperature, because of that change in selection. So the other thing that we can do is we can look at disparity through time. So these plots are kind of it gives you a rough idea of kind of the amount of variation that you would predict to see at that point on the phylogeny. So I'm just going to zoom in on one really quick. And I just really want to bring your attention to all these like peaks and valleys that I have numbered here. And so these are showing kind of spikes where suddenly we're seeing like an increase in the amount of variation that these limb bones were exhibiting at that time. And then you have points where that would drop down. But again, we can overlay our glacial cycles onto this, which is where it gets really cool. So you can see that we have this initial peak falling right in the middle of a glacial cycle. And so then we have a decline, and then we have another peak in the middle of a glacial cycle. And this other peak here is actually during the Younger Dryas, which is pretty cool, which is another cooling event that was not associated with a glaciation. Yeah.
Just wanted to jump in real quick and see if you can start wrapping it up and make sure that we get the audience to ask their questions on Facebook. Thank you.
Okay, yep, I am almost there. So just to summarize what we think has happened and how glaciers played a role here. So we think somewhere around 2.2 million or 540,000 years ago, during an interglacial period, the last common ancestor of martens would have come into North America. They would have spread across North America. But then as glaciers expanded around 540,000 years ago, they ended up isolated into those two regions I was showing earlier, and one in the Appalachians and one in the Rockies. And then they would have diverged morphologically and genetically. And they did this because they were experiencing very different environments in each one of these regions. So in the eastern US, they may have been experiencing more deciduous trees, and in the western US, more coniferous. And so now their limbs look different from one another. And as Blaine pointed out earlier, they do hybridize. So likely what happened if they're hybridizing today, then they probably were capable of hybridizing in the past. So another thing that probably occurred then is as glaciers retreated, they were able to expand into those new biomes, come into contact, and hybridize again. And that this pattern of expansion and hybridization and then being isolated probably happened about 6 different times throughout their evolution because of glaciers retreating and expanding over and over again, which is just really cool. So just to kind of sum that all up, yes, these limbs seem to be evolving, and at least in their shape, as an adaptation because of glacial cycles and the craziness that ensues in environments and climate as glaciers are expanding and retreating. But the super cool part of this, I think, is that the size of these bones and the shape of these bones is evolving differently. And they're independent of one another, and therefore are being selected on by different environmental factors, which is just so cool. So here's my fossil. I promised one fossil in this talk. There are extinct martens in North America that were around up until the end of the Pleistocene. So they're actually one of the examples of a small mammal that went extinct when the megafauna did. And so what I'm wanting to do next, and what I've been kind of working to do, is look at this extinct marten, which is the noble marten, and its range is highlighted there in pink, and try to get an idea of how it differs in its skeletal shape, but also how it differs genetically. And so this is where my ancient DNA training kind of comes in, and I've been working towards getting these guys sequenced for a long time. I feel like every talk I give, I'm like, I'm still doing it, but we're getting there. So I just wanted to thank everybody who worked through all this and all the museums. These projects like this are just pivotal around museum collections and everything that they have tucked away there. So yeah, and so now I'm happy to take all the questions you have for me.
Thank you, Leah. That is really cool research.
Thank you.
And one of the things that I immediately wonder is whether or not there have been other studies that are showing similar patterns with maybe rodents or something like that, you know, Appalachian versus Rocky Mountain populations.
Yeah, so that slide of animals that I showed early on with the voles and the squirrels, those animals have been studied at population levels, not using the same methods. Most of them are genetic studies, but showing that they clearly diverged from one another around that same time. They were in these 2 different regions. So that definitely seems to be a pattern amongst lots of animals, mammals, and birds, at least. But my research is pretty unique in that I'm looking at kind of a population level. Most of these methods are used at like a species level, so you're comparing across multiple species. But these are kind of one of the first studies that's looking at, you know, the same individual's morphology and genetics at that population level.
Excellent. All right, David, do we have some Facebook questions coming in?
We sure do. We've got a bunch coming in already, and I'll just remind everybody that now's the time. If you have questions, go ahead, put them in the Facebook comments, or once again, if for any reason you can't comment on Facebook, head over to the Great Fossil site, Twitter or Instagram, and send us your questions there. I'm going to start with question. We've got a couple questions about DNA research that you talked about.
Okay.
So Grant asked, and you did mention, I know that at least one portion of the genetic material you studied was mitochondrial. Grant was asking, are you using mitochondrial or nuclear sequences? So
far I've used predominantly mitochondrial. The reason for that is mitochondria, there's lots of them in the cells, right? And so you're more likely to get the DNA out of specimens that are older. So some of the specimens that I've been sequencing are from like the 1800s. And so I've been working with mitochondrial DNA because it's more likely to be captured. That being said, the ancient DNA methods are getting to be amazing. And so I am slowly progressing into seeking out some nuclear genes, but everything I've worked on for this project has been mitochondrial.
Gotcha. And then we had another genes question from Chelsea, who asks, have you repeated this with other genes? So are there different genes that show potentially different results?
Yeah, so I have also done what's called a concatenated tree, where you actually combine multiple genes into one phylogeny. And then as you're making that tree, you can tell the analysis to, you know, give different of those priors to each gene. And then you end up with one phylogeny, but it's taken into account multiple genes. So I just showed the cytochrome B gene here, but I do have other mitochondrial genes as well that I combined into one tree, but they give pretty similar results.
All right, we have a couple of people that have asked more about the noble Marten. If you could elaborate a little bit more on that and how it relates to the extant taxa.
Yeah, so that's a great question. So the noble Marten is found predominantly in the same region where you find Martes carina. So it was during the Pleistocene, it was overlapping in the same geographic space as Martes carina. The big debate there is if it is its own species or if it is the same as Martes carina, but just exhibits a different morphology. So it may be one of those examples of plasticity like we showed there. So I have an abstract out there, but not a paper yet, actually looking at isotopes in these guys and some preliminary genetics as well to try to get at that answer as to were they occupying different habitats than Martes carina, and are their genes different? So,
are they easy to distinguish based on morphology?
Their cranial morphology, I think, is their postcranial is statistically it separates out pretty nicely from Americana and carina, but I don't know if I just held a humorous if I could be like, yeah, that's definitely correct.
Grant has a question, a very specific question about a very specific chart that you showed.
Okay.
In the graph you showed with where you numbered the peaks.
Yeah.
Throughout the glacial periods, Grant asked if the number 1 peak is the MIS6 glacial.
MIS6 glacial. Oh, that is very specific. So that one is going to be the Illinoisan glaciation. And I honestly can't remember if that's 5 or 6, but it's Illinoisan is what that one is.
All right. Hopefully that is enough for Grant to link it up to numerically.
I hope so.
And then Ethan.
Oh, sorry.
Yeah, if I could just jump in, Leah.
Oh, sure. Go ahead, Chris.
You know, it's kind of following up on Grant's question. 540,000 keeps popping up as kind of a significant date here. You know, what is going on there? That's the start of all this stuff. And is there some sort of speciation event? Is that do you have any feelings on kind of what is driving, you know, kind of the separation of these species with that particular date? Or is it something that's kind of a wiggle room in terms of the date and maybe a little bit earlier or a little bit later?
Yeah, so where that 540 is coming from is that initial phylogeny that I had, right? And that's where that time calibration put me at. But yes, there is an error bar around that, right? Because that's how phylogenies and time calibration works. But when, it'll take me forever to slide back to it, but when you look at that error bar, the interesting thing is that I've looked at both ends of it and they still place it in the middle of a glacial period. So this whole theory still holds even if I'm kind of wiggle room within that error bar, which is kind of cool.
Is there anything specific to that glacial period that it's worse or it's
so that's that one is part of our pre-Illinoisan like series of cycles, and from poking through the literature, that one there's not a ton of information because it gets wiped out by then the more recent glacial events. So I haven't found anything that says specifically like, oh, this catastrophic thing happened here or anything like that. But it seems to be kind of part of those first cycles that we're starting up.
Maybe we have it backwards. It's the pine martens came in during that glaciation, and that's what sets it apart.
Yeah, and I think that's probably likely because our last common ancestor of these guys is in Asia. And so we probably had an interglacial period that allowed them to just hop on over, and then the rest is history, if you will.
Great. Grant has confirmed that MI6 is the Illinoisan, or
Great.
MIS6. MI6 is James Bond thing. So, actually, I should also make the point. So this is Grant Zazula we're talking about. Both Grants have also left comments in the comments here about collaborations. So if people want to talk to Leah about collaborations, please contact Leah. I'm sure she'd be happy to receive those.
Yeah, I had my Twitter handle up at the beginning. I can.
Oh yeah, go ahead and put that on the screen.
Back up if people want to do that, or you can find me I think I'm so new that I don't know that I'm on Midwestern's page yet. But if you need contact info, Blaine or David could probably send it.
I do think that there must be cave specimens that are out there from Western United States that would be Pleistocene, that maybe haven't been looked at yet for potential DNA, because some of those drier caves have that potential. And then, as I mentioned before we started, I think there is a cave or two that we could look at for you here in the Appalachians that might have some good material.
That would be awesome. Yeah, that'd be great. I've looked at specimens that Berkeley has, which would have been collected out west. All the nobilis specimens came from lava tube caves, and they've been of good quality to work with, obviously, aside from just being old. And then I know that there's been a few other Martens from out west that have been described recently that I need to get my hands on once those pubs come out. So.
We do have some other questions, since we can get through a few more before we wrap up. One more from our Facebook comments is from our friend Ethan, who asks, do the populations in different biomes differ in morphological variability as well as shape?
Oh, that's a good question. So going back to my PC plots in my head, I don't know that they do really. When I'm picturing the morphospace in my head, they're pretty comparable to one another. So I know that animals from broadleaf and boreal forests tend to overlap a little bit more than those from coniferous forests, and we kind of see that in the plots that I put up earlier. But as far as like broadleaf occupying like a huge space within that plot, not really. But I haven't looked at just like, variation as a number specifically.
Gotcha. And then we have a few questions from our students who are here with us. So going back way back to the beginning of your presentation, you mentioned the parasites being used studying these species. Alexis asked, is there a particular parasite that you focus on when trying to determine those species of Martes from each other?
That is a good question, and that is not in my research. That is me quoting someone else's work. So I don't recall which one they looked at, but I believe they were looking at the genetics of those and then how those were branching and whether or not that was comparable to the branching that we see in Martens.
Gotcha. All right. And now we've got another question from Julian. Julian asks, do boreal versus broadleaf trees affect the behavior of different populations in terms of climbing or hunting, or is there something specific that these bone differences, these morphological differences might be displaying?
Yeah, so I think differences in tree type is definitely going to influence how arboreal they are. But also what we're looking at is differences just in overall vegetation density in these forests. So broadleaf forests are what, we're all pretty familiar with, up in the Northeast US, and then also in the Appalachians, right? And so you have that kind of a lot of denser undergrowth than you would see in boreal forest biomes. And so the animals are maneuvering around in that undergrowth differently than they would in more cleared forests up north. So that is definitely going to influence then how they're locomoting and therefore the shape of their bones.
David, I'll jump in and ask one here on another graduate student of ours, Kelly Lubbers, asks, since the pine marten species could potentially hybridize or does hybridize today, is there any evidence of that with the genetic material? And so would you actually see that in the genetics as well?
Yeah, so you do. There's another wonderful scientist, Josie Colella, whose research is focused on the hybridization and parsing that out in different populations. And so she studies a lot of the extant populations today and is actively collecting samples from trappers and stuff. But in order to parse out whether you're looking at a hybrid or not, you have to look at both mitochondrial and nuclear DNA, because mitochondrial DNA is just inherited from the mother. So you're only going to get half of the signal. So if you only look at mitochondria, then they would all look like separate species. But if you also then look at their nuclear, they may be showing up mitochondrial as carina, but nuclear as Americana. And that tells you that you're looking at a hybrid.
Thanks.
We've got just a couple more. Let's see if we can get through them. Christiane asked, is there a reason why you looked at only the long limb bones? Do you think you'd see more evidence for selective pressure by looking at carpals and tarsals as well?
So yes, potentially, because those are going to be moving around as they're grasping things and walking. The difficulty in working with small mammals is that they're small. And so when I was trying to put landmarks on things and surface scan things, when you're working with a carpal of a marten, it's like this big. And so surface scanners don't particularly like that very much. And this is where getting into CT scanning is really where looking at those small things becomes more possible. So future work, future access to CT scanners, I guess. But that's why I started there.
We have one more from David Carney, also a graduate student of ours. Do you see the phenotypic plasticity of the limb lengths in only the eastern, broadleaf, and boreal populations, or is that plasticity also observed in the Western populations?
So when I did this study, they were all kind of combined together. And so you're seeing plasticity across the both species. But if you look at just Martes americana, you do see that difference in boreal forests versus broadleaf forests. I haven't teased apart in that same way Martes carina quite as much because I just don't have the number of specimens there. And so I could look at that and maybe get a similar signal, but my my power in my statistical analysis isn't quite great, so I don't necessarily trust it.
I have one more, David. And it's basically, you know, this idea of using the modern distributions to interpret where an animal prefers to be. And, you know, whether or not something like a pine marten might also still do well if it lived in southern parts of the Appalachians, and how well we can really know whether or not they're limited.
Right. So what it appears happened, at least in the Appalachians, is that the forest biomes that you see martens in today were the forest biomes, or were similar to the forest biomes that used to be in the Appalachians. And that it seems like martens just followed their preferred habitat. Mm-hmm. So it's possible that they could come down in the Appalachians and do okay, but they didn't want to, I guess. Yeah, right. They seem to continue to track as that glacier retreated. They tracked at least what was comparable to the biome that they were experiencing in the Appalachians in the past.
Right. I mean, I guess one of the things I would always wonder is, you know, was there a preferred prey that they were following? You know, maybe they really like to eat phenacomes, this particular vole that moved to the north.
Yeah.
And in those cases, we don't always know.
Yeah, and I think that's where kind of doing these same studies on multiple species within a habitat is really where we'll start to get these more broad picture evolutionary events, I think.
Wow, just fascinating to me.
Thank you.
Thank you.
It looks like we're out of questions for now. So thanks for everybody who participated.
Yes. And thanks again, Leah.
Yeah, thank you.
All right. Well, we'll just go ahead and stop there then and see everybody next week. So I guess
right. Chris, turn off the live stream.
See you, everybody.